Hi friends, in this tutorial we are going to discuss about the enzymes in DNA replication. DNA is a genetic material in all organisms from bacteria to blue whale. The basic essential function of a genetic material is the ability to make copies. DNA replication is a process whereby DNA makes exact copies of itself. Here, a strand serves as a template for the synthesis of daughter strand. There are approximately 20 different enzymes involved in DNA replication, and this is a process of high precision and high fidelity, as any mistake in this process can be disastrous and will be passed on to the next generation. This meticulous process is carried out by enzymes and proteins. Here we are going to discuss in detail all the master molecules behind this process. These are the major enzymes and proteins involved in DNA replication: DNA A protein, helicase, SSPs, DNA topoisomerase primase, DNA polymerase, DNA ligase, TAS protein. In the case of eukaryotes, the proteins are almost similar. There is a slight difference in DNA polymerase. There is alpha and delta, and also in termination process telomerase is involved. And these are the proteins involved in initiation process, DNA A protein up to chirase, and these are the proteins involved in elongation process, that is from primase to DNA ligase, and termination process is carried out by TAS protein and telomerase. And we will be discussing in detail function of each and every protein. This is a double-stranded DNA molecule. There is a specific sequence called origin of replication where the DNA replication starts. The first step in DNA replication is the recognition of ORI by a protein called as DNA A protein. And this is a DNA A protein which recognizes the ORI region of the double-stranded DNA molecule. Binding of this DNA A protein stimulates the denaturation of that particular region, or breaking of hydrogen bonds occurs at that region. DNA A protein or initiator protein, approximately 20 to 50 monomers, binds to the ORI and causes denaturation at that region. ATP is required for the process. This is followed by the binding of enzyme called helicase or TNAP. Helicase binds near the replication fork and actually unwinds the double helix in opposite direction. That's why DNA replication is called as bidirectional. The third enzyme that binds to the site is the SSPs or single strand binding proteins. These proteins actually prevents the rejoining of this single-stranded DNA molecule and prevents the formation of hydrogen bonds between these strands. And secondly, it also protects the single-stranded DNA molecule by degradation by nuclease enzyme. Far away from the replication fork, another protein will bind to the site, that is topoisomerase 1, DNA topoisomerase 1, that is making transient cuts in the DNA molecule, and that is actually relieving torsional stress or negative supercoiling due to the unwinding of the DNA molecule by helicase enzyme. Now the enzymes in the elongation process. First of all, there is no non-DNA polymerase that is capable of initiating a DNA synthesis. A primer is required, and that is synthesized by the enzyme primase. And this primer actually provides a free 3'OH end so that a DNA polymerase can synthesize from that end. And the enzyme for DNA polymerization activity is a DNA polymerase. DNA polymerase elongates the strand, adds nucleotide from the 3'OH end of the primer region. This is a template-directed enzyme that adds complementary nucleotides to the free 3'OH end. And this continuously synthesized strand is called as a leading strand. The other strand, which is synthesized as short fragments, is called as a lagging strand. Here many primers are required, and that is also done by the primase enzyme. In prokaryotes, both lagging and leading strands are synthesized by DNA polymerase 3, whereas in eukaryotes there is a slight difference. Leading strand is synthesized by DNA polymerase delta, and lagging strand is synthesized by DNA polymerase alpha. After the synthesis of lagging and leading strand, there are certain problems. First is their RNA is present in these regions, and that should be replaced. And there should be a joining between these molecule, and that is done by the ligase enzyme. Ligase enzyme catalyzes the joining of DNA strands by phosphodiester formation, and ATP is required for the process. The second problem is the removal of RNA, and that should be replaced with DNA molecule, and this process is carried out by another DNA polymerase which is called as DNA polymerase 1. And DNA polymerase has an additional function which is a 5'3' exonuclease activity, and by using that function it will remove this RNA and replace it with DNA molecule. Then there is a problem in the case of eukaryotes. The chromosome is linear. The problem lies here. This much of DNA will be lost during each replication, and this is referred as end replication problem. But there are certain mechanisms to overcome this problem. In the case of prokaryotes, two circular DNA molecules will be present, a parental strand and a daughter strand, and that will be interlocked, and this is unlocked by an enzyme called topoisomerase II, which is capable of causing double-stranded cuts in a DNA molecule. Then the termination is by a protein called as TES protein that binds to specific regions called termination regions, and this forms a hairpin-like structure, actually blocking the activity of DNA polymerase. But in the case of eukaryotes, there is an end replication problem that should be sorted out, otherwise each replication will lead to loss of DNA molecule. And this is done by continuously repetitive sequences, which is called as telomeres at the end region of the chromosome. The process is sorted out by an enzyme called as telomerase. This is a telomerase enzyme. If you find this video useful, please don't forget to share. You are with biologyexamsforyou.com. Thank you so much for watching.